Mindscape. Today we are going to know about this amino acid which have a non-polar hydrophobic R group. Now all the structure of these amino acids are same, only the R group is different. Now let's see the glycine. It is smallest and HRL with R group H here, the non-polar but slightly flexible due to small size. Then alanine, here we can see a methane group CH3, simple methane group which is hydrophobic. Then valine, it consists CH in bracket CH3 twice. It is a branch chain, bulky, hydrophobic. Then leucine, CH2 CH, CH3 and CH3. It is a branch chain and common in alpha helix. Now isoleucine, we are drawing it CH in bracket CH3 CH2 CH3. So isomer of leucine and branch chain. Methionine, CH2 CH2, here sulfur group is present and CH3 is present, so it contains sulfur but still non-polar. Also starting codon AUG amino acid is present. So proline forms a ring with amino group which is CH2 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 and NH. So cyclic structure, it is rigid, break alpha helix and secondary amine. So phenylalanine, here we can see the R group CH2 C6H5. Here benzene group is present. It is aromatic and large and non-polar. Now tryptophan, it is partially polar but grouped as a non-polar. So here the R group CH2 is indole ring, double ring structure with nitrogen. It is aromatic and bulky and weak polar but generally considered non-polar in hydrophobic terms. Now let's know a mnemonic for knowing this. So here's the mnemonic. Great ape proudly travel places in mighty land. So here G for glycine, A for alanine, proudly for proline, travels for tryptophan, vast for valine and places for phenylalanine, and N for isoleucine, mighty for methionine and land for leucine. So polar uncharged amino acid. This group 2 amino acid are serine, cysteine, threonine, tyrosine, aspartate and glutamate. The side chain in the group possesses a spectrum of functional group. Most of it have at least one atom, for example nitrogen, oxygen or sulfur, with electron pair available for hydrogen bond to water and other molecule. Now let's see the chemical structure of this group 2 amino acid. Serine, cysteine, asparagine, glutamate, threonine and tyrosine. Two amino acids, serine and threonine, it contain an hydroxyl group, that is an oxygen atom bonded to a hydrogen atom represented as OH. Then tyrosine possesses hydroxyl group in the aromatic ring, making it a phenol derivative. Now hydroxyl group in these three amino acids are subjected to important type of post-translational modification, phosphorylation. As we can see here, the other non-standard amino acid like methionine, cysteine contain a sulfur atom. Unlike methionine, sulfur atom, however, cysteine sulfur is very chemically reactive. Asparagine first isolate from asparagus and glutamate both contain amide R group, and the carbonyl group can function as an hydrogen bond acceptor, and the amino group, that is NH2, can function as an hydrogen bond donor. Now acidic amino acid, there are two amino acid in this group. They are aspartic acid and glutamic acid, and each has an carboxylic acid on the side chain that give it acidic or proton donating properties. In an aqueous solution at physiological pH, all the three functional group on this amino acid will ionize and thus give an overall charge of -1. And that in this ionic form, that amino acid are called aspartate and glutamate. The chemical structure of group 3 amino acid are given here. The side chain of aspartate and glutamate can form ionic bond, that is a salt bridge, and they can also function as an hydrogen bond acceptor. Many protein that bind the metal ion, metaprotein, for structure or functional purpose, possess metal binding site containing aspartate or glutamate side chain or both. Free glutamate and glutamine play a central role in amino acid metabolism. Glutamate is most abundant in neurotransmission in the central nervous system. Now the three amino acids in this groups are arginine, histidine and lysine. Each side chain is basic, that is it can accept a proton. So the lysine and arginine both exist with an overall charge of +1 at a physiological pH. The guanidinol group in arginine side chain is mostly basic for all the R groups. And mentioned in this diagram, aspartate and glutamate, the side chain of arginine and lysine also form an ionic bond, and the chemical structure of group 4 amino acid are given here. So amidazole side chain of histidine here present here, we can see this. It allow it to function in both acidic and basic catalyst near the physiological pH values. So none of other standard amino acid possess this important chemical property. Therefore histidine is an important amino acid that most often makes up the active site of the protein enzyme. And the majority of amino acid in group 2, 3 and 4 are hydrophilic, that is water loving. As a result, they are often found clustered on the surface of the globular protein in aqueous solution. Amino acid by their various chemical function, for example carboxylic, amino and R group, and can undergo chemical reactions. And the peptide bond and cysteine oxidation are of particular importance because it affect on the protein structure. Amino acid can be linked by a condensation reaction in which the OH is lost from the COO group, that is carboxylic group of one of this amino acid along with hydrogen bond from the amino group of the second, forming a molecule of water and leaving the two amino acid by an amide, and it is called a peptide bond. At turns of 20th century, German chemist Emil Fischer first proposed this linkage together of amino acid. When individual amino acids are combined to form a protein, their carboxylic and amino group are no longer able to act as an acid and base. Since they haven't reacted to form this peptide bond, therefore the acid-base properties of protein are dependent upon the overall ionization characteristics of an individual R group of the component amino acid. So amino acid joined by a series of peptide bond are said to be constituent of peptides. So after they are incorporated to an peptide, the individual amino acid are referred to as an amino acid residue. Smaller polymers of amino acid, they are fewer than 50, are oligopeptides, while larger ones which are more than 50, they are known as the polypeptides. So hence a protein molecule is a polypeptide chain composed of many amino acid residue. When with each residue join the next by an peptide bond, and the length of the different protein range from a few dozen to 1,000 of amino acid, and each protein contain different relative property of the 20 standard amino acid. So what do we learn? What is an amino acid? An amino acid is an organic molecule that is made up of NH2 amino group and an acidic carboxylic group COOH. An organic R group that is unique to each amino acid. The term amino acid is a short form alpha amino, that is the carboxylic acid, and each molecule contains a central carbon atom and called alpha carbon, to which both an amino and a carboxylic group are attached. The remaining two bonds of alpha carbon atom are satisfied by an hydrogen atom and an R group. So amino acid function as a building block of a protein. Protein catalyze the vast majority of a chemical reaction that occurs in a cell. They provide structure element of a cell. They help to bind a cell together into a tissue. In human body, there are 20 amino acids that function as a building block of protein. 9 of these amino acids are essential. They must be consumed in diet, while 5 are considered non-essential. They can be made by the human body. So the remaining 6 protein building amino acids are conditional, being essential only at a certain life stages and in certain disease stages. The essential amino acids are histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan and valine. The non-essential amino acids are alanine, asparagine, aspartic acid, glutamic acid and serine. The conditional amino acids include arginine, cysteine, glutamine, glycine, proline and threonine. So some authorize recognize a 21st amino acid, cysteine, which is derived from serine during the protein biosynthesis. Now, what is the difference between the standard and non-standard amino acid? They are the classifications of amino acid based on the polarity or the distribution of the electric charge of an R side chain group. So 20 amino acids that function as a building block of protein are classified as standard. The non-standard amino acids basically are amino acids that have been chemically modified after they have been incorporated in a protein. So they can also include amino acids that occur in living organism but are not found in protein. Among this, gamma carboxylo glutamic acid and calcium binding amino acid residue found in the blood clotting protein, that is the prothrombin. The most important post-translational modification of amino acid in eukaryotic organism, including the human being, is phosphorylation, in which a phosphate molecule is added to the hydroxyl portion of a serine, threonine and tyrosine. The phosphorylation serves a critical role in the regulation of protein function and cell signaling. So amino acid, any group of an organic molecule that consists of a basic NH2 and a COH group and a R group, that is a side chain. Now let's know a mnemonic for the uncharged polar side chain where serine, threonine, cysteine, asparagine and glutamate and threonine. So here the mnemonic is small tall cat snapped quietly yesterday. Thanks for watching. Do like, share and subscribe.